This is Kazufumi Honda from Nippon Medical School, who has been working on developing biomarkers for cancer screening using minimally invasive methods. In this video, I would like to talk about how we can use aberrant truncation of the C-terminus apolipoprotein A2 homodimer for early diagnosis and risk stratification of pancreatic cancer. Here you can see the survival rate of Japanese patients with pancreatic cancer at 1 to 5 years, and the colors show each clinical stage, stage 1 to stage 4. As you can see, pancreatic cancer is a refractory type of cancer, and early detection is essential for improving survival rates. The most reliable treatment for pancreatic cancer is surgery. However, surgery is very often not indicated because almost all patients are already at an advanced stage of the disease at the time of diagnosis. If we can detect pancreatic cancer at stage 1, we will be able to increase the survival rate of patients by about 40 times compared with patients diagnosed with stage 4 cancer. And this is the goal of our research, to find a biomarker that can be used to identify pancreatic cancer through a minimally invasive means like a blood test. To identify patients with early-stage pancreatic cancer in the general population, we need an efficient enrichment method for high-risk individuals. This method should use minimally invasive biomarkers, be low cost, and be in the form of a blood test. After we identify patients who are at risk, we can use high-resolution imaging, such as the methods listed here, to find pancreatic cysts or pancreatitis or precancerous lesions. To find the most efficient biomarkers, we used mass spectrometry to profile the alteration of peptides in the bloodstream and identify those that have different profiles between pancreatic cancer patients and healthy subjects. Then we used proteomic analysis to identify aberrant processing of C-terminal amino acids in patients with pancreatic cancer. As you can see, circulating apolipoprotein A2 mainly exists as a homodimer in the bloodstream. There are 3 isoforms that have different amino acids at the C-terminal end. The C-terminal end of the heavy type is ATQ-ATQ, and the light type is AT-AT, and the intermediate type is ATQ-AT. We found 2 interesting things. First, aberrant processing of C-terminal ends of the circulating apoA2-isoforms were found in patients with pancreatic cancer. And second, there were 2 unique alterations of processing patterns, hyperprocessing and hypoprocessing. Significant reduction of intermediate type apoA2-ATQ-AT was observed in the bloodstream of patients with pancreatic cancer as compared to healthy subjects. To easily detect the processing patterns of apoA2 isoforms in the patient's blood, we developed a sandwich ELISA assay kit in collaboration with Toray Company. This is a schema of the ELISA system for measuring apoA2 isoforms. As you can see, there were high correlations between the results of mass spectrometry and ELISA. This slide shows the clinical utility of the apoA2 isoform ELISA kit for detecting patients with pancreatic cancer. We collected plasma samples from patients with pancreatic cancer, other cancers, and benign diseases from 7 hospitals in Japan and used them to validate the clinical usefulness of the ELISA kit. As you can see, the ATQ-AT isoform was significantly decreased in patients with pancreatic cancer of all stages in comparison with healthy controls. And this graph shows the receiver operating characteristic curve analyses. The area under the curve of the ATQ-AT isoform was higher than 0.92 for all stages of pancreatic cancer. We proposed a collaboration between the National Cancer Institute Early Detection Research Network and the National Cancer Center Japan, which allowed us to assess their pancreatic ductal adenocarcinoma reference set in order to confirm the ability to distinguish between pancreatic cancer and healthy controls. As you can see here, a significant reduction of the ATQ-AT-isoform was recognized in stage 1 and stage 2 pancreatic cancer patients as compared to healthy controls. In addition, the blind test by NCI-EDRN also confirmed that the area under the curve of apoA2-isoform is higher than that of CA19-9. Also, as shown in this figure, the area under the curve of the combination assay with apoA2 and CA19-9 was significantly higher than the single examination of CA19-9. This shows that the combination assay could significantly boost the area under the curve from 0.783 to 0.879. The areas under the curve of ATQ-AT were high not only in pancreatic cancer patients, but also in high-risk individuals, such as those with IPMNs and chronic pancreatitis. We found that these abnormal processing patterns were inversely associated with the exocrine function of the pancreas. The pancreas has a kind of carboxypeptidase, which are enzymes that cleave the C-terminal ends of peptides in the pancreas. It's possible that those enzymes are aberrantly cleaving the C-terminal of apoA2 according to the exocrine function of the pancreas. This is a diagram showing the hyperprocessing and hypoprocessing patterns, both of which are prevalent in patients with pancreatic cancer, as shown in this figure on the left. In addition, these processing patterns were recognized not only in pancreatic cancer patients, but also in individuals at a high risk for pancreatic cancer, such as those with IPMN and chronic pancreatitis, which corroborates what we said before. However, these processing patterns were not observed in other gastroenterological cancers. Next, we evaluated the risk stratification of pancreatic cancer using apoA2-isoform ELISA of pre-diagnostic samples obtained from the EPIC study, which was organized by the WHO International Agency for Research on Cancer. The EPIC study is one of the largest cohort studies in Europe, with more than 500,000 participants from 10 European countries, and followed for almost 15 years. And they have collected a large number of pre-diagnosis samples. We used the apoA2-isoform ELISA kit to evaluate alteration of apoA2 processing in EPIC samples. All double positive cases seen with our method and CA19-9 testing were diagnosed with pancreatic cancer within 5 years after blood collection. To validate the clinical utility of apoA2 for pancreatic cancer screening in the general population, we carried out experimental pancreatic cancer screening of blood samples for apoA2 at Kobe University and 7 screening centers in Japan, starting in 2015. First, the subjects underwent a blood test for identification of apoA2 and those who tested positive were recommended by their doctors to undergo imaging examinations for pancreatic disorders. This slide shows the association between apoA2 and patient characteristics. When screening the general population, apoA2 were significantly associated with abnormalities on ultrasound imaging and a history of DM and pancreatic disorders. In the multivariate analysis, we found that the odds of having lower levels of apoA2 was 3 times as much as those that didn't have lower levels. This slide shows the breakdown of high-risk diseases found by blood tests for apoA2 We enrolled 5,120 participants from an asymptomatic population in the Kobe Cancer Screening Network. apoA2 blood tests identified 84 subjects as positive for the marker. 54 of these 84 subjects underwent CT, MRCP, or EUS, and of those, 26 showed abnormalities on imaging of the pancreas. 18 malignancies were found among the 54 cases who were considered at a high risk for pancreatic cancer. This indicates a positive predictive value for distinguishing high-risk individuals of 33.3%. Compare this to the use of imaging modalities to identify cystic lesions in the pancreas in an asymptomatic population, and the positive predictive value ranges from 1.2 to 3.5%. IPMNs are classified into two groups according to their malignant potential: IPMN with high-grade dysplasia and IPMN with low-grade dysplasia. The high-grade dysplasia type is considered a non-invasive cancer and should be surgically resected. On the other hand, the low-grade dysplasia type is not considered an indication for surgical resection. However, it should be strictly followed up by imaging because the 5-year risk of developing into PDAC ranges from 2.2 to 8.8%. Since these people have a significantly higher chance of developing pancreatic cancer than the general population, if these patients are identified using non-invasive methods and then strictly followed up with imaging modalities, then the early stages of PDAC can be detected and early curative surgical resection can be performed. In order to confirm the detection performance of apoA2-isoforms for IPMNs, we collaborated with Professor Strobel, Professor Buchler, and Dr. Felix of the University of Heidelberg and confirmed the distribution of apoA2-isoforms in the bloodstream of patients with IPMNs and healthy controls. This slide shows the detection performance of apoA2-isoforms for IPMN. This method has a high sensitivity for detecting patients with IPMNs with LGD, HGD, and early-stage IPMN associated with cancer. The area under the curve values are shown here and are all significantly higher than CA19-9, which are shown in red. This slide shows the sensitivities and specificities of apoA2-isoforms and CA19-9 to detect patients with IPMNs and PDAC. As you can see, the sensitivities of apoA2-isoforms were significantly higher than that of CA19-9. In particular, the sensitivity of apoA2-isoforms for detection of IPMN with HGD, which is considered carcinoma in situ, was 70.6%, and this was significantly higher than the sensitivity of CA19-9, which was only 14.5%. Currently, we are conducting studies at several hospitals to confirm the clinical utility of apoA2-isoforms for the enrichment of PDAC and high-risk individuals. This is a schema of the expanded prospective experimental screening for PDAC. In this study, 10 to 20,000 subjects over the age of 50 will be enrolled from screening centers in Kagoshima, Kobe, and Hokkaido, Japan. Subjects who are positive for apoA2-isoforms will undergo dynamic CT. Finally, we will calculate the detection rate and positive predictive value of PDAC and high-risk diseases. We aim to follow all the subjects to observe their survival rate. Additionally, we will calculate the relative risk of pancreatic cancer using the biomarkers, and we will estimate the sensitivity and specificity of these biomarkers from follow-up data in the Cancer Registry of Japan. We received IVD approval of apoA2-isoform from the Ministry of Health, Labor and Welfare of Japan on June 8, 2023. So, we'll be able to use the apoA2-isoform in clinical settings for detecting patients with pancreatic cancer. Here's a brief summary. First, apoA2-isoform is a potential biomarker not only for the early detection of pancreatic cancer, but also for stratification of high-risk individuals. Second, we have developed in vitro diagnostics using this biomarker, which can be used in clinical settings in Japan. I would like to thank the many collaborators and investigators shown here and many others for their role in this work. These studies were supported by the Japan Agency for Medical Research and Development.